Hello, everyone, and welcome to today's webinar, Use of Echo-Acoustic Liquid Handling in Functional Genomic Screening of Retinal Ganglion Cells. I am Michelle Ashton of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Labcyte. To learn more, please visit www.labcyte.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speakers. Derek Welsbie, the Assistant Professor of the Department of Ophthalmology at the University of California, San Diego, and Iain Russell, the Senior Product Manager at Labcyte. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Our speakers will now begin their presentation.
Thank you, Michelle, for the kind introduction there. Before I get started here, what I'd like to do is give you a very brief introduction into Labcyte as a company. Labcyte was founded almost 20 years ago and was the first company to use sound energy to transfer fluids for both drug development and life science research. The key benefit to our acoustic transfer technology is the ability to transfer low nanoliter to low microliter volumes of fluid with extremely high precision and accuracy. In 2011, dynamic fluid analysis, a proprietary technology, was introduced, and that enabled our echo liquid handling systems to automatically adjust for properties such as viscosity and surface tension across a broad range of fluid types without any user intervention. And I'll go into this in a little bit more detail on a later slide. This technology has greatly expanded the echo liquid handler capabilities for use in many different applications and has led to a strong adoption of the echo liquid handler in fields such as genomics and synthetic biology. Supporting higher level automation of our customers' workflows, Labcyte offers a complementary robotic platform that enables the integration of the echo liquid handler with additional devices for a completely hands-free experience. With adoption of acoustic liquid transfer technology in the genomic space, there did come a need for a system that could build larger volumes faster. This need was filled in 2013 with the launch of the Echo 525 instrument, and this has become our preferred system for genomic applications where larger reaction volumes are preferred. Not shown on this slide is the very recent acquisition of Labcyte by Beckman Coulter Life Sciences, now bringing two leading technology platforms under the same roof. Okay, let me tell you a little bit about the technology that provides the foundation for the echo liquid handler. The echo liquid handler uses sound energy to move liquids from a source well to a destination well that is positioned above the source in an inverted orientation. At a high level, the process is relatively simple. Sound energy created by an acoustic transducer is focused at the surface of the fluid to create fluid droplets of a highly controlled volume. These droplets are then ejected from the surface with sufficient velocity to reach the target well, which can either be dry, as in the example shown in the cartoon, or already containing fluid. Depending on the instrument, the droplet will either be a 2.5 nanoliter or a 25 nanoliter droplet in volume. To transfer larger volumes, the system simply transfers multiple droplets in quick succession. The systems typically transfer anywhere between 200 and 500 droplets per second. Because the transducer can be aligned with any source and any destination well, the system is capable of any well to any well transfer. Key benefits to this technology is that there is no physical contact with the sample in the well, so that maintains sample integrity. The energy is very low energy. The droplet size is highly consistent, either 2.5 nanoliters or 25 nanoliters in volume. And it is rapid and flexible, transferring 200 to 500 droplets per second. Now, with anything, it's not quite this simple. And so I do want to dig into some of the key underlying technologies that we have driving this acoustic transfer. To ensure the high accuracy and precision of the liquid transfers, Labcyte uses a process we call dynamic fluid analysis to interrogate the content of the source well. This utilizes the ability of the transducer to not only emit sound energy, but also detect the reflected sound energy or echoes, much like sonar or medical ultrasound. As an example of this ability, what you are looking at is a representative cartoon of the detection of sound energy reflected from the bottom of the plate, the bottom of the well, and the surface of the liquid. Each of those reflections has a time and amplitude component, which change based upon the composition of the liquid. The important benefit of this technology is that it enables the Echo liquid handler to support a very broad array of different fluid types, including cell lysates, various reagents at varying concentrations, even those including glycerol, and various biomolecules, all without the operator having to make any adjustments to the instrument either prior to or during the transfer. Now I'm going to switch gears slightly and introduce the Echo product line. This slide provides a little bit more detail into the products offered by Labcyte. The Echo liquid handler product line is composed of three main instrument families: the Echo 525, the Echo 550 series, and the Echo 650 series. These instruments differ in terms of their speed and types of volume they excel in transferring. The Echo 550 and 650 series instruments offer that 2.5 nanoliter resolution droplet that I described previously and are a recommended line of instruments for drug development. The Echo 525 instrument, on the other hand, offers a 25 nanoliter droplet resolution and is ideal for workflows such as genomics, where building larger volumes is required. All these instruments are supported by a catalog of acoustically qualified microplates offered by Labcyte. What is not shown on this slide is a variety of different software applications, each tailored to meet the types of workflows of interest to our customers, and the services that we offer to ensure that you are able to focus on your science at hand. So what are the benefits of acoustic liquid handling? The benefits of acoustic liquid handling really fall into four main categories. First one is maximizing the lab budget. Secondly, increasing data quality. Thirdly, speeding up assay design and execution. And lastly, the ability to fully automate complex workflows. In the next two slides, I'm going to dive into two of those benefits in more detail. When you take into consideration the ability to miniaturize assays using the Echo liquid handler, our customers have been able to achieve up to 100-fold reduction in reagent usage. That also translates to less sample consumption, which can be particularly important for those with limited or particularly valuable sample types. With the ability of the Echo liquid handler to perform any well-to-any well transfers extremely rapidly, in many cases, complex workflows can be significantly sped up. Although this is only one example, in the example shown here, an 18-hour workflow involving sample pooling and enzymatic assemblies was reduced to only 3 hours using the Echo liquid handler, saving 15 hours of time. What I've given you is an overview of the Echo liquid handler family. All these instruments are extremely simple to use, are ready to go out of the box, can be easily integrated into larger automated solutions, and share the same underlying acoustic contact-free transfer technology. With that, I want to thank you for all your attention, and will now turn over to Derek, who's going to present on some of the work that he's been doing using the Echo Liquid Handler. Thank you.
So thank you so much for joining us. Today we're going to be talking about functional genomic screening in retinal ganglion cells. So first, a disclosure. Nothing in this presentation should be construed as an endorsement for Labcyte or its products on behalf of the UC Regents or UC San Diego. Now, my lab has been interested in developing novel therapies for a group of eye diseases called optic neuropathies. These are all characterized by the death of retinal ganglion cells, or RGCs. But since this might be a diverse audience, I figured we'd spend some time on background. So shown here is a cross-section of a brain and eyeball cut right down the middle with an enlarged view of the retina and a cartoon of a single retinal ganglion cell. Light hits the eye and is focused onto the retina, onto various spots on the retina. That light is then processed by photoreceptors into an electrochemical signal that's then sent over to the retinal ganglion cells. The signal is actually processed by the retinal ganglion cells and then transmitted down the optic nerve to higher order centers in the brain, as shown here, and that results in vision. Now, I've shown one retinal ganglion cell, but in reality, there's about 1 million of these lining the human retina, with each one representing about a pixel of vision. And of course, if there's 1 million retinal ganglion cells, then if you looked in the optic nerve, there'd be approximately 1 million axons. Now, shown here is a typical fundus photo of a normal, healthy optic nerve and retina, showing the optic disc, which is the confluence of all the retinal ganglion cell axons before they exit the eye as the optic nerve. So then the question is, what is glaucoma? So in glaucoma, there is a death of these retinal ganglion cells. I'm showing you now the same fundus photo, but with a little cartoon of retinal ganglion cells, what they would look like. Again, I'm showing you 12, not 1 million, just to make things more clear. In glaucoma, these axons get damaged at the optic nerve head at a structure called the lamina cribosa. In response to that injury, there's a retrograde injury signal that propagates up the axon and triggers an active genetic program of axon degeneration and cell death. And this starts off in these peripheral or somewhat peripheral retinal ganglion cells, but it continues, it propagates, it progresses, and it moves closer and closer to the center of the retina called the fovea. And as you lose these axons, combined with the changes in the optic disc structure, you get this characteristic shape of the optic nerve called cupping, which is sort of very characteristic of glaucoma. Now I'm going to show that same animation, but this time I'm going to show a visual field test, which is what a patient would do when they're being evaluated for glaucoma. So that little black spot in the visual field here is normal. That's the physiologic blind spot. And keep in mind that everything is reversed so that the inferior visual field is processed by the superior retina. So at first there is this damage to the optic nerve, and that triggers the degeneration and cell death. But initially you have more retinal ganglion cells than you need, so even though you've lost retinal ganglion cells, we don't see that as a loss of vision on a visual field test. However, as this progresses more and more, you eventually dip into your reserve of retinal ganglion cells, and it starts to functionally disconnect entire swaths of retina from the brain, leading to these visual field defects. And again, this progresses. As you lose more and more retinal ganglion cells, it gets closer and closer to the center of vision. And some patients, sadly enough, even despite treatment, can lose all of their retinal ganglion cells. And also because of the limited capacity for adult neurogenesis, this vision loss is essentially permanent. And so in 2019, we can't restore vision, and the only thing we can do is to lower eye pressure. In summary, glaucoma is an axon injury disease of retinal ganglion cells, and the only treatment is to lower eye pressure. And it's interesting, right, because I give this introduction to glaucoma, there really is very little mention of eye pressure. You know, almost 50% of patients never have an elevated eye pressure, and the reason it's so central to glaucoma therapy is simply because it is our only modifiable risk factor. So what our lab has been very interested in is, you know, can we identify a neuroprotective strategy that might complement IOP lowering? And the idea behind a neuroprotective strategy is that it might interfere directly with that retrograde injury signal in response to that axon injury, or with the active genetic programs of axon degeneration and our cell death. And by directly protecting the retinal ganglion cell, we think we can hopefully improve the options for glaucoma patients. So, you know, over the years, lots of people have tried lots of neuroprotective strategies. So what we thought we might try and do is set up a comprehensive and unbiased screen of potential drug targets. We developed a phenotypic screen using primary mouse retinal ganglion cells. To interfere with gene function, we used the genetic technology of RNA interference initially, mediated by small interfering RNAs or siRNAs. And each of these would knock down a target gene. If the target gene is involved in cell death, then its knockdown should promote survival, as diagrammed here in this cartoon. To do this, since we were going to work with primary retinal ganglion cells, we needed a way of delivering RNA to the cell in a fashion that was sort of amenable to high-throughput screening. Fortunately, we came across this magnetic nanoparticle called Neuromag from Oz Biosciences. And what we do is we couple the nanoparticle to nucleic acid, and that's as simple as simply mixing them together, and then adding them to the media. When the cell culture plate is placed on a magnet, which we actually just keep in our incubator, it brings down the magnetic nanoparticles, probably increases their local concentration at the bottom of the well, and leads to uptake of the nanoparticles. And for instance, if we put in a GFP mRNA, we get really, really nice transfection efficiencies. And the system is very amenable, since it's simply a mix and add type system, it's very amenable to high throughput screening. Shown here is the bright field image of our primary retinal ganglion cells and the GFP fluorescence after we've transfected them with GFP mRNA. Now, the system works great for delivering RNA. It does not work well for delivering plasmid, probably because of the additional barrier of having to get to the nucleus. So then the next challenge was we had to obtain siRNA libraries. And so we worked with NCBI, to get that. So we had multiple siRNA libraries. And they came in plates where the siRNA was approximately 10 micromolar. So we had to make intermediate plates. And for that, we made intermediate plates using these V-bottom polystyrenes. Now, we would typically fill them using the combi, just bulk liquid dispensing. We'd dispense 20 microliters of Optimem into all the wells, and 100 nanoliters of Neuromag. And again, that's all mixed together and just bulk dispensed. We would then transfer the siRNA library using the Labcyte Echo. Now, typically, we screen at 1 nanomolar siRNA, and our source wells, the ones from NCATs, were coming in at 10 micromolar. Since at a 96-well plate we would have a final volume of 100 micromolar, we needed to do approximately a 1 to 10,000 dilution of the siRNA, which meant that we would use the Echo to transfer 10 nanoliters. Now, fortunately, that's right in the sweet spot for the Echo. So we would just go through and use the Echo to transfer over the siRNA to these pre-dispensed V-bottom intermediate plates. And we would fill them up. We'd leave the edge columns open for controls. Once we had finished with our intermediate plates, we would turn to the assay plates that will actually be used to culture the cells. And these are standard tissue culture coated NUNC plates. We would coat them with PDL and laminin. And then we would transfer the library from those V-bottom plates using a liquidator, just essentially a 96-well or 96-head multi-channel pipette. And then the idea would be to have the cells go on top of that in a reverse transfection paradigm. So to get the cells, we would use P0 wild-type black 6 pups, and we would immunopan them using anti-Thi1 coated plates. From there, we would seed these into the multi-well dishes, again using a combi. And I'm showing the screen for 96 wells, but we've done this with 384-well plates and 15 36-well plates. And essentially the workflow is the same. So we use the combi to add the cells on top. Now remember, there's already a transfection mix sitting in each of these wells, with each well getting a different siRNA. Now, either because of the isolation of these retinal ganglion cells, which necessarily exotomizes them, or something about the way we are culturing them, we get pretty profound death over the next 3 days, which sort of makes sense, right? If you exotomize a retinal ganglion cell, it does die. That's kind of the idea behind glaucoma. So we're using this natural cell death after isolation as our selective pressure, as our model of injury. Then what we need to do is figure out which wells that we've intervened on that have increased in survival, So we've done this 2 different ways. When we initially did this, we used the Genomics machine, but more recently we've been using the molecular devices Image Express, and we simply have an automated imaging platform where we go through and take pictures of all the wells, and then automated image analysis to quantify the number of surviving neurons. And when we've done our smaller scale screens, this has worked just fine. For larger scale screens, where we're up into the multiple tens of thousands of wells, we have found it easier to adapt the platform to cell titer glow, which is a reagent from Promega. It's an ATP-based luminescent assay. And then we just used a BMG plate reader to quantify survival. And again, if we're doing large screens, we find that to be a little bit easier. So the work started with the kinome. And the reason we started with the kinome was, well, there's a few reasons. First, it's a small percentage of the genome, about 2 or 3% of the genome, so it makes things easier to get started. Second, the kinome tends to be rather bioactive. And finally, if we find anything, it's pretty easy to envision making drugs because of the rich history of medicinal chemistry when it comes to developing kinase inhibitors. So shown here is a histogram of the results from our first kinome screen. So there's just a little under 700 genes in the mouse kinome. We had 3 siRNAs per gene, and shown here is the histogram of survival. As you can see, most siRNAs cluster around the 0 or slightly negative survival, which makes sense. Most siRNAs, when they knock down their target, don't do anything to survival, or if anything, they decrease survival. What we were interested in is what were those siRNAs that are out here whose, when you knock down the target gene, you get an increase in survival, suggesting that that target gene is involved in cell death. The top hit in the screen were the actual 3 oligos against dual leucine zipper kinase, also known as DLK. Very satisfying, but actually the number 2 hit in the screen were the 3 siRNAs against MKK7, which at the time was the only known substrate of DLK. To validate what we were finding on plastic had some in vivo relevance, we turned to Aaron D'Antonio's floxed DLK mice, in which the addition of CRE leads to an incomplete kinase domain and loss of function. So again, given that glaucoma is an axon injury disease, we used the quick and dirty model of optic nerve crush to validate DLK. First, we would intravitreally inject virus that delivers CRE and that transduces retinal ganglion cells into either floxed DLK mice or the wild-type control. We would then wait 7 days to allow the virus to complete its life cycle and for the extant DLK to turn over. At that point, we would then crush the optic nerve mechanically with a 5-second crush. And then after 10 days, given that you've now injured the axons of the retinal ganglion cells, you'll see death of retinal ganglion cells. So we quantify that by staining flat mounts of retina and then doing automated image analysis. Shown here is a retinal flat mount that's been stained for retinal ganglion cells, and it's replete with the normal population of retinal ganglion cells. 10 days after crush in control mice in which DLK is present, you see the sort of typical 2/3 loss of retinal ganglion cells. However, in mice which carry the floxed DLK allele, the addition of CRE quite robustly increases the amount of survival that you see 10 days later. Now, it was nice because this work was actually independently validated by Joe Lucock's group at Genentech. So they also showed that the loss of DLK leads to increased survival of retinal ganglion cells in vivo. And actually, if you went 18 weeks out, you could see that that survival was still maintained, which was nice, that we were not just delaying cell death, but preventing. And what this taught us was that our in vitro platform of using mouse primary retinal ganglion cells could teach us something about these in vivo optic neuropathy models. Finally, because of course we are interested in ultimately developing glaucoma therapies to protect retinal ganglion cells that have been injured in a glaucoma model, we turned to the rat laser model, which you laser the trabecular meshwork, raise the eye pressure, and measure the number of surviving retinal ganglion cells either in the retina or optic nerve about 6 weeks later. And we used a small molecule inhibitor that has activity against DLK, packaged it in such a way that it would release over approximately the 6-week period when injected intravitreally, and showed that that small molecule inhibitor improved survival. Now, it was interesting. When we were doing this work, we noticed that the effect of the drug, tozasertib, which again is an inhibitor against DLK, would give much greater survival than when we genetically knocked out DLK in vitro, as shown here. And it made us think that perhaps the drug was hitting some other target in addition to DLK that was improving our activity in vitro. So to identify that missing target, what we did is repeated our kinase screen, looking through the kinome, but this time we started with DLK already knocked down, and we repeated. Now, of course, DLK doesn't come out anymore because DLK is already knocked down in every well. But now what came out were the siRNAs against LZK, or leucine zipper kinase. We made the leucine zipper kinase knockout mouse, the conditional knockout, bred it to the conditional DLK, and what we showed in vitro when we added adenovirus to transduce primary retinal ganglion cells with CRE is that the addition of CRE would give you a small increase in floxed DLK retinal ganglion cells. Remember what I said before, genetic knockout of DLK by itself in vitro doesn't do that much, but it's certainly above baseline. But the knockout of LZK does essentially nothing to survival. Of course, if you knock out both DLK and LZK, we saw this very robust increase in survival. This was done in primary retinal ganglion cells, but we also showed that there was additional survival in vivo in the optic nerve crush model. Now, it turns out that this gave us a nice opportunity because LZK knockout by itself doesn't really do much to survival. I mean, those are the triangles you can barely even see here of the controls. However, if you additionally knock out DLK in the setting of an LZK knockout, you get this very robust increase shown here. So we thought, hey, this maybe gives us an opportunity to sensitize the screen to things that are in the DLK pathway. And that increased signal to noise might allow us to not just screen the kinome, but to screen the entire mouse genome. So we did. We've actually done this now in multiple screens. But shown here are the results of screening approximately 18,000 siRNAs. And these are little mini pools of 4 siRNAs per well. It was satisfying that the well that contains the mini pool against DLK was actually the #1 hit. So the screen in general was working to identify things in the DLK pathway because it identified DLK. But when we went to confirm the other genes that were increasing survival by ordering new siRNAs against those, we really found almost nothing was confirming. The reason for this is likely the lack of specificity of siRNA. So unfortunately, these are pretty non-specific. You know, when you have an siRNA, you've got your 21 nucleotide sequence, which I'm sure matches a single mRNA in the genome, and so that's rather specific. However, these siRNAs also have this 6 to 7 nucleotide seed sequence shown in orange. And because it's only 6 to 7 nucleotides, it's much less specific. And so it binds to the 3' UTR of multiple mRNAs and leads to silencing. So unfortunately, the ratio of things that are non-specific to specific is probably something like 20 to 1 or 100 to 1. And so it really makes it difficult to see effects through the noise. We tried to address this 2 different ways. First was seed correction. We know the seed of every siRNA, and we know that we sample that seed in the context of different siRNA sequences. What we can do is keep track of how that seed non-specifically is affecting survival. And then we can generate a handicap score. So you can imagine for the seed GUAA, GCU, shown here in red, every time an siRNA had that seed, there was this increase in survival. So if we see a target siRNA that we're interested in, and it's got that seed, it needs to have even more than 2-fold survival to be interesting. So we essentially can go through and re-handicap all the siRNAs. The second thing we did to try to deal with this off-target issue was a haystack analysis. So again, we know the seed in every siRNA. Moreover, we know all the potential off-targets that it could be hitting. So what we do is we keep track of the seed, the off-targets, and the effect on survival through every well. And if we've screened enough wells, and we've done screens of almost 60,000 siRNAs, what we can then do is figure out, are there genes that tend to correlate with that seed and with survival? So for instance, in all the wells that have increased survival, we might tend to see seeds that silence BBC3 or PUMA. So this is another way of getting at potential hits. But again, this is sort of bioinformatically deconvoluting the data to try to find something interesting amongst all this noise. It would be much better to have a system where we could screen that simply doesn't have all the noise. And so we'll get into that in just a second. The second issue that we ended up with was we would do these screens and we'd get these very interesting candidates, but then we had to validate it. And again, with all this noise of siRNAs, we weren't going to be satisfied with validations that use siRNA. We really needed knockouts. Now, we were obtaining the floxed mice for a lot of these candidates, like SOX11 or MEF2A, and we would import the mice, they'd go through quarantine, we'd breed them up, we'd eventually have enough mice that we could isolate primary retinal ganglion cells from them, and we could do the in vitro assay by adding adenocre to these conditional knockouts. And if we saw increased survival, as you're seeing here with SOX11 or here with MEF2A, we knew that indeed our results from the siRNA screens were validated. But that was a lot of work. And you know, we have this great high throughput system, but this is as low throughput as it gets. So we needed something different. And that's why we turned to CRISPR screening. So the way this would work is we would provide a guide RNA to various targets that binds to Cas9. Cas9 comes in and using its 2 nuclease domains creates a double-stranded break. That double-stranded break is repaired by the non-homologous end-joining machinery, but because there's no template there, you end up with mutations. And if you've chosen targets that are in the coding sequence of the gene, you end up with frame shift mutations that cause a loss of function. So the strategy was we would isolate retinal ganglion cells from mice that already have Cas9 in every cell. These are Feng Zhang's ROSA26 mice, which have constitutive expression of the strep pyogenes Cas9. And all we would have to do is we don't have to deliver Cas9 now. We're just responsible for delivering the gRNA. But as I showed you before, both with the siRNA data And with the GFP mRNA data, RNA we were pretty good at delivering. So the way we would do this is we would order an oligo against any given target. So for instance, let's say SOX11 comes out of the screen, and we want to validate SOX11. We would order an oligo that has a gRNA sequence targeting SOX11. On the 5' end, that oligo would have the T7 promoter. And on the 3' end, that oligo would have a piece of the gRNA scaffold or the tracer sequence. And because the entire sequence is less than 60 nucleotides, that is a relatively inexpensive primer to have synthesized. And then we would have a second primer, which has the remainder of the gRNA scaffold or tracer sequence. We could anneal these two, and we would transfer them to these low dead volume echo plates, and then use the echo to transfer these gRNAs. Then we would use a PCR machine to do elongation of those annealed primers. And what you end up generating is what's shown up here. You end up generating the entire gRNA sequence with a T7 promoter up at the 5' end. Then what we needed to do was, we run the PCR to the extension, then what we need to do is from that PCR product, use the T7 promoter to do in vitro transcription. We would use the HiScribe Quick T7 high-yield RNA synthesis kit. Again, we would use the echo to transfer the right amount of kit, the right amount of our PCR template that we had just made, and we would do the in vitro transcription. And what that would generate is lots of in vitro transcribed gRNA. We would then purify that in vitro transcribed gRNA and use that for the transfection. And although initially in our screens we were doing things in which the neuromag was transferred via the combi in bulk transfer, now we were transferring the neuromag with the echo and doing the preparation of the transfection mixtures all with the echo software. And that seemed to be, that's worked pretty well. So then as we generate lists of potential targets, all we do now is order a new 55 nucleotide sequence gRNA, in vitro transcribe the gRNA, and use that to test whether there is relevant biology. So does this whole system work? Well, the first thing we had to show was that the system wasn't toxic. We were delivering, these RNAs that were, approximately 100 nucleotides long. And so using the primary retinal ganglion cells, we showed that in general, there was no non-specific activity of the gRNAs. They weren't toxic. Next, we made a series of gRNAs against DLK, right? Because that was our positive control. And we tested different amounts. And what we showed is that these gRNAs, when you put them into primary retinal ganglion cells isolated from wild-type mice, do not increase survival because there's no Cas9 to do CRISPR editing. Control gRNAs don't increase survival even when given to Cas9 RGCs, but DLK targeting gRNAs, when selectively given to Cas9 expressing retinal ganglion cells, gave this very nice increase in survival. And one thing that we noticed was it didn't really matter which exons we were targeting. We saw a little bit of variability, but maybe not as much as one would expect in the amount of survival. The second thing that was maybe a little bit interesting is that when we combined gRNAs, we really didn't see much improvement. It sort of maxed out, as shown here. When we combined, we didn't see much improvement. Actually, I think I misspoke. Here we've used the DLK Assay pool to knock down DLK, and we're combining it with the LZK gRNA. The one I just described is actually here where we're knocking down DLK, and LZK is knocked down with an Assay pool. We've also done it where we've knocked down both DLK and LZK only with gRNAs. Next, we wanted to look at off-target cutting. So we chose a gRNA sequence that would disrupt a restriction site. So if we were getting good cutting by our Cas9, we should not get good cutting by the VGL2 restriction site because the mutagenesis should disrupt recognition by VGL2. And indeed, we could show that if we amplified the DLK locus, that the addition of the gRNA targeting DLK would create this new band that's not digestible with VGL2. However, when we looked at the various off-targets, we really weren't seeing much, maybe a little bit for one of them, but in general, we were not seeing much off-targeting, which was satisfying. We looked at whether we needed to have homozygous or heterozygous mice, and it turns out it really did not make much of a difference, that if you put the gRNA into either Cas9 heterozygous RGCs or Cas9 homozygous RGCs, we essentially got very similar levels of gene editing, maybe with some trend towards homozygous being better. If we looked at the mutagenesis that we created, we saw that when we put in 2 gRNAs, we would often see inversions of the intervening sequence or deletions of the intervening sequence. But only occasionally would we see individual mutations where only one site but not the other is being mutagenized, as shown in construct 9. If we put in a single gRNA, we saw the typical cutting where the mutagenesis are sort of typically small deletions, and they're centered around the 3rd and 4th nucleotide just upstream of the PAM sequence. And using this technique, we were able to produce mutagenesis that was approximately 50% of the population of primary retinal ganglion cells. So we've used that technology now to validate various target genes. I think I have a slide on that here. So what you're seeing here are Cas9 retinal ganglion cells isolated in vitro, in which we have created in vitro transcribed gRNAs against various targets, ATF2, MEF2A, SOX11, and Jun, and shown that when we add these guys individually, we don't see much increase in survival, but when we knock them out in a combinatorial fashion, we see relatively nice survival in vitro. And we've done sort of optimizations of dose, and it really does tend to be that for us, about 30 nanograms of sgRNA or synthetic guide RNA is sort of the sweet spot for producing knockouts. But now that we know that, what we can do is we can consider screening. We have this echo-based system where we can do all this synthesis of gRNAs in sort of small scale per well, but that means we can do a lot of different wells, and we can transfer things using the echo and do screens. So to prove that a screen was doable, we actually used the DLK gRNA in RGCs that are lacking LZK. And if we put in control guides, we see that we have a very consistent lack of survival or death at baseline. But when we put in various wells in 384 well format, guide RNAs that have been in vitro transcribed using the ecosystem, we see increases in survival. And although there is some noise, there's such clear separation between background that this makes for a good screen. So we are now in the process of screening a library that targets the mouse kinome to see if there are kinases that we missed with siRNA-based screening, either because of the noise or because we need a knockout rather than knockdown to see the biology. And we're hoping that this new system allows us to identify new kinase targets. Using our prior system, we've developed this model of cell death in which injury leads to activation of DLK, LZK, and various downstream targets to mediate cell death. In conclusion, we've developed a high-throughput method for functional genomic screening in mouse primary retinal ganglion cells. We started off with RNAi-based screening, but now have moved to CRISPR-based screening. And we've used a variety of machines, but one that has been really enabling for us is the Labcyte Echo, simply because it allows very small volume transfers. This has been part of a long-standing collaboration with Don Zach. So, of course, I want to thank Don and the rest of his lab, including Cindy, John, who is now at Labcyte, Yukon, who made a lot of the gRNAs, Vinny, who did some of the conceptualization of the gRNA method, Pingu and Katie, who did some of the work creating these. We have a high-throughput screening course here at UC San Diego with Carl Wallen and myself, Risa, Amit. Amit should get a lot of credit here. Amit's the postdoc in my lab who's done a lot of this work, and also a collaboration with Robert Weinreb. Thank you very much for listening, and I'll now take some questions.
Thank you, Derek and Iain, for your informative presentations. We will now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. The first question is, most vendors sell guide RNAs in which the tracer RNA and the CRRNA are two separate molecules. Have you tried that, and how does it work?
So we have, actually. What I showed was where we were creating an in vitro transcribed 100 nucleotide-ish gRNA, the kind of chimeric gRNA, in which the tracer and the CRISPR RNA are fused into one. When we've compared that versus some of the commercial vendors' 2P system, we see almost a log loss of activity. So for whatever reason, in our system, when we break this up into two pieces, it has not worked nearly as well. Now, we've tried the Dharmacon system, their CRRNAs and their tracer RNA. We haven't necessarily tried all the different vendors out there, but just when we have tried it, it has not worked particularly well, and we really don't know why that is, because obviously in the endogenous system in bacteria, it is a 2P system. So really, one would think it should work, but in our hands, not as well.
Thank you. Our next question is, how do you choose your guide RNA targets?
There's a variety of criteria that go into choosing our guides. So, we have used, there's a ton of websites out there that'll kind of predict off-target effects. We, I think, have sort of vectored towards using the Broad Institute's on and off-target ranker. That gives us a general list of gRNAs. Now, because we are doing T7-based transcription, ideally our gRNAs would start with 3 Gs. So it'd be the motif GGG, N17, NGG. However, we've realized that we really don't need to start with 3 Gs. We can start with 2, and actually we've even started with just 1, and it's perfectly fine for T7 transcription in our hands. Also, because we don't want to throw out some of those really highly ranked gRNAs, we've made gRNAs that are not the 20 nucleotide spacers, but only 19 or 18 or 21, and those all seem to work equally well. So you can move that first G anywhere in the first 4 nucleotides, and it seems to be just fine. We try to make sure that the gRNA targets common exons. Ideally, we want the CRISPR cutting site between that 3rd and 4th nucleotide to fall within a restriction site, only because it makes the downstream quantification of cutting so much easier. Because as I showed, all you do then is a PCR to flank that region, digest the PCR product with the restriction site, and there's an inverse correlation between the amount of restriction cutting and the amount of CRISPR editing. And so that works pretty easily. The gRNA sites tend to be between sort of the 5th and 65th percentile of this coding sequence, although that's not our rule. That comes from the bioinformatics software. We do like to have gRNAs that target both mouse and human, although I haven't talked about it today. We've been now doing this work in iPS-derived human retinal ganglion cells, and so it's nice when we have one gRNA that we can cross-shop across the 2 different platforms. And I think that's the last thing we've done recently. There was a paper that came out at the end of, I think, last year by Graf et al. That showed that there are a few of these motifs like GCC or TT in the distal part, the part next to the PAM, where you want to avoid those for good gRNA design. So we've been avoiding them. We're not sure if it makes a difference, but we've been doing that. In general, we pay a lot more attention to bioinformatic predictions of off-target cutting than we do of on-target cutting, because we don't see so much variability in the on-targets, but we know that the off-targets, that was our whole history with siRNAs, that can be a problem. So that's sort of our priority.
Great, thank you. Multiple papers have shown that chemically modified guide RNAs have improved editing capability. Have you tried these?
So we have. Now keep in mind, we are very limited in what we can use because we use the T7 polymerase to make our gRNAs. So a lot of these chemically modified gRNAs come from chemical synthesis. And then you can put in all sorts of modified bases. However, for us, the only one that's really compatible with T7 is the 2' fluoro gRNA. And so we did that. We transcribed these 2' fluoro gRNAs, and we saw absolutely no difference in editing. If anything, maybe it was a little bit worse.
Our next question is, does the echo require calibrating for each fluid type?
So the echo does not require any calibration by the user, irrespective of the type of fluid that they're using. Labcyte is calibrating all the echo liquid handlers in the factory before they go out, and the calibrations capture very, very broad fluid categories. So, for example, that might be surfactant-containing fluids, buffers alone, or perhaps buffers that have are viscous in nature because of glycerol or some other viscous material being in there. Those high-level categories are selected by the user, and then it's the dynamic fluid analysis technology which enables the echo to adjust on the fly for the nuances of the particular fluid that is being transferred. So it's very simple to use. It's simply the user just needs to understand the high-level category in which the fluid lies. They select the appropriate calibration that is offered on the instrument, and then they start the fluid transfer process.
Thank you. Our next question is, can you use any type of plate with the echo?
That is a good question. For the destination plate, the answer is yes, provided that it is ANSI/SLAS compliant. So the instrument gripper supports any plate types that are automation friendly, ANSI/SLAS compliant. We even have customers who are using slides or Petri dish type of consumables where you can purchase carriers for those. Now on the source plate side, so this would be the microplate from which you are transferring your fluid, we do require that you use an echo qualified plate. These are plates that Labcyte provides or offers, and we go through a lot of effort to ensure that those plates are acoustically compatible and will provide the optimal fluid transfer when used with the echo liquid handler technology.
How does the ecosystem ensure there is no cross-contamination?
This is, of course, a very important aspect of any liquid handler. The echo liquid handler, if it wasn't apparent in my presentation, does not use any tips or pins or anything that physically touches the fluid. So it's purely the sound energy that is generating those droplets and ejecting them out. So with that, you completely eliminate any potential for cross-contamination that is resulting from tips involved in the transfer process, which can be a fairly major source of cross-contamination depending on the type of system and the fluids being transferred there. The other aspect there is the dynamic fluid analysis is constantly adjusting the parameters of the acoustic transfer process to ensure that the transfer is optimal and that we maintain that high degree of accuracy and precision for the volumes that are being transferred.
Thank you again, Derek and Iain, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Labcyte, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.